right, so now we're in the physiology part. Uh, the heart is a muscle. If you remember, there's three types of muscles. There's smooth. There's smooth. There's cardiac, and there's skeletal. Cardiac muscle is kind of like skeletal muscle, but there are some key differences. One being that cardiac muscle is involuntary, whereas skeletal muscle is voluntary. But another difference is that in between each cell, you have these intercalated discs. So you could see it like between this cell and this cell. Muscle cells are elongated. They're not like these circular things. They're elongated cells that are able to contract. So they have these intercalated discs, and then if you look inside of them, there's like these gap junctions. So what this is doing is it's letting the electrical impulse jump from one cell to another cell really quick. In fact, those electrical impulses, we call them action potentials. Right? So an action potential is, think of it as the electrical impulse that's going through your heart. So heart's different. Heart has to beat all at the same time. So the atria contract simultaneously, and then the ventricles contract simultaneously. So it's like when you look at the heart, the top part of the heart beats, then the bottom part of the heart beats, and then the whole heart relaxes. So you could view that kind of like in three stages. Top part of the heart beats, then the ventricles contract, and when the ventricles contract, the atrium are relaxed. So atrium contract, ventricles contract, whole heart relaxes, then it starts over. When the atrium or when the ventricles contract, they all have to do it at the same time. Right? And when you look at skeletal muscle, you don't have to use all of your fibers when you're picking up a light object, for example. You're not using all your fibers. As things get heavier and your muscles have to do more work, it recruits other muscle fibers. So not all your muscles have to go. It could go at like 50% or 25%. The heart has to go at 100% or it's not going to go. So that electrical impulse goes through the entire top part of the heart at pretty much the same time. And so that's why we have these intercalated discs. So there's like a order that this action potential goes through. And this slide represents one of the possible quiz questions. So this is the cardiac conduction system of the heart. Right, so it's pretty much just listed. It's already listed out here. So, I mean, that part's kind of easy. But it goes right, like it starts here at the right atrium. Inside the right atrium, that's where your pacemaker is. So the pacemaker is the sinoatrial node, SA node. So you start with the SA node, and then the action potential you see like with these purple arrows, it's going to the other atrium, it's going to the left atrium, and it's going down to the right atrium. So the pacemaker, the SA node, that's where the action potential starts. It's going to cause the two atrium to contract. And then that action potential moves down to this node right here. It's kind of at the junction between the two atria and the two ventricles. It doesn't look like it necessarily on this slide, but it's kind of close to these two atria and where these two ventricles are. And so we call that the AV node. If the SA node doesn't work and stops working, the AV node can kind of pick up and start taking over as the pacemaker. It's not that you're going to be healthy. You're not going to be running any marathons or anything like that, but it will work enough to keep you alive. But anyway, from the SA node, we go to the AV node, AV being atrioventricular, atrial for atrium and ventricular for ventricles, right? AV node. Then look at it here. It's going to drop. It's going to drop down into the ventricular septum. So this muscle here, this border in between the two ventricles, we call it the ventricular septum. There's also an atrial septum. It's like right here. You can't really see it that well, but you can see the ventricular septum, right? So from the AV node, we're going into the bundle of His. So right here, there's like a there's a bundle of this nervous tissue. And then you see in number 4, it's going to split into the left and right bundle branches. So this is to have some kind of blockage in here is something that if you deal in cardiology, it like happens. It's one of the many things that happen, right? A bundle branch block, right? So it'll something's not letting the impulse pass through here. Or you could find a case where people will it's too active. Like this is acting like a pacemaker. Let this person. So you can find a case where it's acting like a pacemaker. And the bundle branches are too active, and it's putting the heart into tachycardia, right, making it beat too fast. But anyway, left and right bundle branches, and then we'll follow one of these branches down. It goes down to the apex of the heart, and then around the other side of the ventricle. So all these 5, and these are called Purkinje fibers. So the cardiac conduction system, or the conduction system of the heart, is just these 5. It's in this order: SA node, sinoatrial node, atrioventricular node, the AV bundle, the bundle branches here in the septum. They become bundle branches, and then they come down on the apex. And become the Purkinje fibers. So that's it. It's just kind of like it's listed here. But I just wanted you to also envision what's going on. This is like the way that the action potential, where it starts and where it ends. If you follow it through, it's, again, top part of the heart, the atria are going to contract, then the ventricles are going to contract. And then when the action potential gets to the end of the Purkinje fibers, there's a relaxation period. And when you look at the heart, it's like, top, bottom, relax. Top, bottom, relax. So that's it. Since we're looking at this, no, it's okay. We'll cover it. Okay. This, we're not going to go over it like strictly, but you need to know how a heart cell or heart fiber contracts. And it's not much different from regular muscle physiology. I have a video where I believe I wrote on it, so it's kind of showing you. But before we get into it, there's some thinking how to explain it. All right. The question is, what makes a heart cell or heart fiber, or any muscle fiber actually, what makes a muscle fiber contract? So whenever you go to contract a muscle, what's actually happening to that cell? Well, the cell's shrinking. It's a long rectangular cell. That's what all your muscle cells are. These really long rectangular things, and then it's able to shrink down and then go back, right? It's able to contract and go back. Just like you learned about in AMP1. Um, what makes that happen is different electrolytes, different minerals. These minerals that we use a lot, we call them electrolytes. And so you see here, there's three we're talking about: sodium, potassium, and calcium. So sodium is, these minerals, they like to hang out either in the cell or out of the cell. So if I were to talk about sodium, I know sodium is extracellular. Sodium always hangs out outside of the cells. That's where it likes to be. Potassium is an intercellular cation. Intercellular meaning that it likes to hang out inside of the cells. So generally, when you find potassium, you find it inside of cells. When you find sodium, you find it outside of the cells. The reason I called it a cation, all of these are cations, is because it's positively charged. So just like if somebody were to speak about chlorine, you'd say, tell me something about chlorine in the body. Well, it's an extracellular anion, meaning that it likes to hang out outside of cells, and it's an anion, meaning that it's negatively charged. Right? So all the electrolytes, all the minerals actually in our body are charged. They're either positive or negative. So sodium is extracellular. Potassium is intracellular. So we're not talking about the membrane potential here. We're not talking about like the voltage. We're just gonna like take these three steps. So the first thing that's gonna happen is that sodium. So right now there's a balance. Before we even get to number one, there's a balance. There's potassium inside the cell and there's sodium outside the cell. If you're looking at things that are like positively charged, they're equal. So yes, there's more potassium in the cell and there's more sodium outside the cell. But if we're just looking at things that are positively charged, they're equal. So what's going to happen now is we're going to throw it off. We're going to send all of the sodiums into the cell. And now all the positive charges are in the cell and not outside of the cell. So that's what number one is. So you had an equal number of positively charged ions or whatever, positively charged things inside versus outside of the cell. Now the sodium all rushes into the cell. So sodium rushes into the muscle cell. That's going to cause the muscle cell to depolarize. You've thrown off the balance. It's no longer equal positive in and out of the cell. Now all the positives in the cell. We call this depolarization. That word comes up a lot. So, I mean, there it is right there, right? So the cells, the muscle cell become depolarized. That's going to cause them, it's going to cause them to contract. So depolarization refers to the electrical imbalance or the electrical signal, right? Contraction, the word contraction, that's just the muscle physically contracting. It's going to contract because of depolarization. We want to hold that depolarization for a while. We want to maintain it. You don't want to just depolarize, contract, and then immediately relax. So you're going to hold this state of depolarization. That's what the calcium does. The calcium is going to start to trickle in. What happens is that sodium and potassium, they don't like to hang out in the same place. They work together a lot. So they're like, I always say they're like a married couple. Like they like to, they have common goals and they like to work together, but they just don't like to be in the same room. So whenever sodium enters a room, potassium's like, yeah, I got a thing to do, and it leaves, and vice versa. So what happens when sodium rushes in, potassium's starting to leave. Like some of the potassiums are like, yeah, it's awkward in here, I'm going to leave. So what happens is a calcium starts trickling in to keep all of the positives inside of the cell, right? So when all the positively charged things are in the cell, that cell is going to be depolarized. So calcium is coming in to kind of hold that depolarization. Then eventually enough potassium leaves, right? A few trickle out, but then eventually they all just make their way for the exit, and all the potassiums leave the cell. And so what they've essentially done is they've changed places, right? All of the sodiums came in, and then all the potassiums went out, and they swapped places. So when all the potassium ions leave, we call that repolarization. It's repolarized. So depolarization, technically what it is when you've thrown off the membrane potential. You've thrown off this balance of positively charged ions inside the cell versus outside the cell. You might have had this last semester, but if you're like me and like every other student, you weren't trying to remember it, even if you had it. You're just trying to get through the test. So here it is again. So, but these words, depolarization and repolarization, we use these words are going to be used for this this chapter, and it's used practically. So, depolarization, that's because sodium rushes into the cell. Plateau, holding that depolarization, that's calcium trickling into the cell. Repolarization, potassium's leaving the cell, like quickly, right? And so that's what they're kind of saying here. They're talking about these voltage gated fast channels. All that means is that you imagine that you have like a cell and there's like a membrane, like a wall around the cell, and the gated channels, the channels are doors. So a door opens, something rushes in. Another door opens, something rushes out. That's what these channels mean. Right? So, the cell membrane is kind of like a wall and a door. If you think about the wall in your house, it's made from two layers of drywall, right? And then there's like studs. So it's like wooden studs and then drywall is attached to it. The drywall is like the phospholipids. There's two layers of them. So it's on both sides. And then in the middle, you have like proteins and stuff. That would be like the wood. But then your walls, a lot of them have like doors and windows, right? So those are like the channels or transport proteins, whatever. But the doors are like the channels. So depolarization, repolarization. This is I don't know how I don't know what to call it. It's the, I guess it's how muscle cells contract, how an action potential moves through an individual muscle cell. So this explains a couple of things. One is like the importance of calcium. Like we were talking about last or a couple chapters ago, how your body regulates calcium with like PTH and calcitonin. There's a reason for it because we need calcium in our blood. We need calcium in our blood to make our muscles, particularly the heart muscle. We need the heart muscle to operate, to function properly. You'll see when IV bags are given, they're sodium chloride. And they kind of match the level of sodium chloride in your blood, right? So we already know that sodium and sodium chloride, those are two important electrolytes. But potassium is another one that's given to people that are dehydrated. But potassium, it's never just hung as like an IV bag. it comes in after. So you'll piggyback it. It's separate. It's made by the maybe the pharmacy will put it together or something like that, right? Because you can't just give potassium intravenously to people because you're gonna mess them up if you give the wrong amount. Potassium is really important to this whole action potential, right? So if you were to give too much, which happens, if you were to give too much potassium to someone, it's going to kill them. It's going to seize their heart, right? And too little potassium in the blood would also cause heart problems. It won't cause it to stop, but it'll cause it to, get like atrial fibrillation or maybe ventricular. I don't remember which one. All right, you want to know what's on the test. This, you have to know the 3 steps. I mean, no point in putting this down exactly as it's written unless you understand exactly what that means. But essentially, you have sodium rushing into the cell that causes depolarization, potassium trickling into the cell that holds it, plateau, potassium leaving the cell that's going to repolarize it. And then before the next heartbeat, the sodium and the potassium switch places again. It's weird. So you have, like, take sodium. It rushes in, and then the potassium rushes out. Why can't they just switch places again and make another heartbeat? They don't. They go back. They switch places to where they were originally, and then the sodium turns around and goes right back in again. So sodium is constantly going in the cell, constantly going out of the cell. Same thing with potassium. It's constantly going out, and it's constantly going back in and then back out again. So you'll learn about it in a different subject, but there's like these sodium potassium pumps. There's like these special, they're like revolving doors that are built into your cell membrane. So when a sodium goes in one direction, the potassium has to go in the other direction. Otherwise, the door doesn't work. So they both got to be at the door, and then it just switches their place, right? But that's in a different class. But you see how much I'm talking about sodium and potassium here. So like these are really 2 important, and calcium, right? So out of all the minerals in your body, I don't know. This is opinion. I think calcium is vastly the most important, and not because of bones, right? But then after that, I would say sodium and potassium, right? So those 3 are like hugely important electrolytes. And when I say electrolyte, I'm just talking, there's minerals that your body uses. Some your body uses often, and some minerals you don't use as much. Iron is not an electrolyte, but iron is a mineral. Iron's very important. You know what it does? It's in your hemoglobin, right? But it's not an electrolyte because you don't use it as often. Sodium, potassium, calcium, these are electrolytes. Like you need it all. You need greater amounts of them. This is and I have this, I believe, on the next slide. I'm explaining what it is, but this is an EKG or an ECG, but everyone calls it an EKG. This is measuring that, what we just talked about. Remember, there's three parts to like a heartbeat: the atrium contracting, the ventricles contracting, and then the whole heart relaxing. That's kind of measured here. There's 3 portions here. There's like this thing here, P wave, and then there's this whole thing here. They call this a QRS complex. I've got it on the next slide. And then the T wave. There's like 3 sections to this. And so what you do is you set up, put little stickers. You'll put 2 here. Depends on how you run it, but like, for what I know, and what I know maybe is like a little bit different, but first we do like a 4 lead. So you'll put 2 here, or you can even put them on the arms, or, shoulders is best. You're sending electrical signals like down this way, right? And then you'll put two down here, maybe on your legs or something like that, but at least below the heart. So this calls like a 4 lead, right? So you got 4 leads on it. Actually, only 3 are working, but anyway, we call it a 4 lead. And what it's doing is it's like shooting, it's looking at the heart. You've got it like coming in diagonally, right? And then usually if you see something like off on this, like that by itself will give you kind of like an EKG. And if something doesn't look quite right, you can add more to it. So then we move to a 12 lead. And what you do is you put a bunch right here. So you're like following the breast, right? And then those are all shooting up at the apex of the heart. So you've got leads here and here up in your shoulders, and then you've got leads down here. So there's 4, right? And then if you want to do a 12 lead, you're going to come in underneath the heart here and just put. You're not putting another 8. I think you're putting. I got to think about it. 6, 5, 4. You're putting like another 4 down there. So anyway, I don't know why they call it a 12 lead. I forget, but this is what it looks like. And then people that are good at this, they could interpret this, and there's a bunch of different readings. There's hundreds of different things that can happen. And I don't know any of them. I really suck at it. I know this ST segment, if it is up a little bit higher, that could mean like a heart attack, like an MI. But there's like 100 different ways to diagnose a heart attack. If you have like a blockage, like a pulmonary embolism, like this thing right here, see this QRS, it might be like elongated out a little bit. So there's things like this, dehydration, like this T wave here, instead of being like a hill, it's more like a triangle, right? Like they're peaked. But again, like I don't know how to like. I've set up a bunch, but I'm not good at reading them. I can read the basis. I'm really good at that. Like when it's just a running straight across, I'm like, all right, dead. I'm pretty good at that one. I know tachycardia is just going like this up and down. That one's pretty easy. A bundle branch block, for example, you'll see. I don't remember how it goes now. Oh, okay. Like you see this whole thing. Like there's P, that's step 1, QRS step 2, G is step 3. You'll see these, right? And then there's like a space right here. What happens is and then you have another one, right? So you see this, and you see another one, and you see another one. The space between each one should be like the same. But when there's something wrong with like the bundle of His or the bundle branches, the space between them gets like more, and then the next one will be even wider. So anyway, I want you to know that. So the P wave is atrial depolarization. So you're thinking of the cells in the atria, sodium is rushing in. Atrial depolarization means the atrium is about to contract. Because of atrial depolarization, the atria contract. That's that first one. So P wave, this is telling me about the atrium. Are they working okay? Are they about to contract? Something's wrong with the atria. Why? Is that because there's like a holdup in the ventricles? Is something wrong with the ventricles and that's causing something to be wrong with the atrium? Whatever it is. QRS, probably the most important out of these 3. Ventricular depolarization. Now the ventricles are going to contract. So remember, atrium contract, ventricles contract, the whole heart relaxes. So when we break it down into the EKG, atrium contract, ventricles then are going to contract because they're being depolarized. And then the T wave is ventricular repolarization, meaning that the ventricles are going to relax. In fact, the whole heart is relaxing. It's just that we're measuring it with the ventricles. So Ventricles are between the atrium and the ventricles. The ventricles are more important. They're pushing blood out, so they're more active. The atria, they also push blood around, but the atria really receive a lot of blood. So that's not a very active process. You're just sitting there and waiting for blood to come into you, right? Whereas the ventricles, they're pushing blood out. So that's why you see more concentration on the ventricles here. So that's what it looks like. There's a whole class on that you could take. If you're going into nursing, you're going to have to learn some of it. Now the EKGs, they tell you at the top what it is. So just like with everything else, take the thinking out of it. Your whole job will be on autopilot. So that's the EKG. You have to know the parts of an EKG and be able to explain what's happening. P wave, atrial depolarization. QRS means ventricular depolarization. It doesn't mean the ventricles are contracting, technically. The depolarization is the electrical part. That's going to cause the mechanical contraction of the ventricles. Sometimes you could have the electrical impulse, but the ventricles are not contracting. So it's getting the electrical signal, but the muscle's not reacting to it. So it's a different question. All right. So let's go back a little bit and review some things that we talked about. So number one, cardiac conduction system. That's a question, and that's an easy question because that's kind of listing. And if you're just memorizing it and spitting it back out, that's fine, right? The SA node is the pacemaker of the heart. Would be nice that you knew that for the future and kind of have an idea of where that is. It's in the right atrium. But, you know, if you just memorize this and spit it back out, you know, that's fine. I like you to understand how this works and not spit it back out. I have, there's lots of videos. I have a video where I'm explaining it with, I have like a rectangle, I think, to represent a cell, and I'm moving the electrolytes in and out, if that helps you. But there's other videos that can explain it. And of course, your book, which we never talk about. You do have a book, and it does explain it. So that's your second question right there. This is your third question. What are the components or parts of an EKG? Right. Don't just say P wave, QRS complex, T wave. What do they mean? What are they trying to say? Right. The P is talking about the ventricles depolarizing, which is going to lead to contraction. And then, you know, the key, it's here. I already said it. So here's your third question. Any questions so far on those three? You guys even here? You are here. Okay, just checking. So you got 3 questions down, hopefully. There's 3. Probably going to be 5 questions on this test. So I've already got 3 down. All right, the 4th one, unfortunately for you, is the most difficult one, but it's like the most important one. I don't think it's difficult. You guys always think it's difficult. It's called the cardiac cycle. It's so if you see, I've got 3 slides just on cardiac cycle. It's not about, in fact, I don't even want you to list it. I'm going to want you to explain it, like write it in sentences. Not to be a dick. Not saying that I'm not, but on this question, I'm not trying to be a dick. I'm just I want you to understand what's going on. I mean, I do not care about 130 milliliters and 25 milliliters. I don't care. It's not important. I want you to try to be able to understand what's going on. If you don't get every single detail and you understand it, it's good. It's not like, I mean, this slide here and your book make it very difficult. It's actually not that difficult. Here, your book puts it into 6 different steps. I'm putting it into 3 different steps. So the 3 steps are the atrium contract. Then what happens after the atrium contract? Because I've been saying it a few times now. Atrium contract, then the ventricles contract. Then what happens? The whole heart relaxes. Nothing's contracting. Atrium contract on the top, ventricles contract on the bottom, second. Whole heart relaxes, third. That's the cardiac cycle. 3 steps. Atrium contract, then the ventricles contract, then the whole heart relaxes. All I'm doing is I'm taking each of those 3 steps and talking about some things that happen. So atrium contract we call systole. Systole, there's different ways to say it. Systole. By the way, don't ever correct people in like anatomy. Like when they say something wrong, maybe they are wrong, just leave it, because there's also a good chance that there's more than one way to say it, and then you're correcting them and you're going to look stupid because their way is also correct. Systole is a good example of that, right? You'll hear people say capillary instead of capillary, and you're like, shut the hell up. It's not even a word. It's a word. Pisses me off when they say it, because as soon as you say capillary, my whole brain starts to search for that word. Wait, what's that word? And I stopped listening. But anyway, systole means contraction. Diastole, here's this word here. Diastole means relaxation. I'm jumping ahead. Systole, or like systolic, diastolic. Systole,
basically like your blood pressure, like the top bottom number.
That's what blood pressure is.
Yeah, the bottom number versus the top number, right? Like
yeah, so the top number, it's seeing what the pressure is here when your ventricle's contracting. What's the pressure on that artery, the brachial artery, when your heart's contracting, when your ventricles are contracting? In that bottom number, they call it diastolic. So it's like, what's diastolic? Your heart. When your ventricles are relaxed, what's the pressure on that artery? Yeah, so that's exactly where it comes from. So yes. So you'll remember the words. It's just, I use those words instead of contract and relax. But if we look at atrial systole, so let's just think about the atrium now. They're sitting above the ventricles. Right, so what's happening is that this is a cycle, right? So there's nowhere that I can really start, because wherever I start, something happened before it. So that's what I'm doing here. I'm saying that your atrium have been filling up with blood. Before, 2 things happen. Your atria fill with blood, and so imagine that like they're filling with blood and there's like a trap door underneath. Like when it gets heavy enough, that door is going to pop open and all the blood's going to drop down. Right? Those are your AV valves, your mitral valve and your tricuspid valve. They're closed, and on top of it, it's all filling, like blood's filling up that space. So it's filling up, and then when it gets to like a certain pressure, those AV valves are going to pop open, and that most of that blood is going to drop down. That's before atrial systole. But when those AV valves open up and all that blood drops down, there's still some blood left over. How much exactly? Who cares? It's around 25 mils, doesn't matter, but there's some blood left over. Now the atrium are going to contract, just squeeze the rest of that blood down into the ventricles. So that's the whole point of atrial systole. The blood actually, most of the blood fell into the ventricles. It just kept filling up the atrium until the atrium got too full and it made the valves open up. And all that blood just dropped in. Not all of it, most of it. The atrium contract to push, I'm not putting it right here, the atrium contract to push the last bit of blood into the ventricles. That's atrial systole. The atrium are contracting, pushing the last bit of blood into the ventricles. Of course, the AV valves are open. That's the valve between the atrium and the ventricle. They're open. Whenever the AV valves are open, the other valves, the semilunar valves, are going to be closed. So if the semilunar valves are open, the AV valves are closed. Whenever one set of valves is open, the other one's closed. And the other thing is, whenever the atrium are contracting, definitely the ventricles are relaxing. They don't both contract at the same time. They can both relax at the same time, but they don't both contract. You're never going to have the atrium, well, you're not supposed to. You're not going to have the atrium and the ventricles contracting at the same time. So when the atrium are contracted, the ventricles are relaxed, the semilunar valves are closed. Now at the bottom here, I've got this thing I'm calling end diastolic volume. So end relaxation volume. Okay, the atrium aren't relaxing here. I'm telling you here that the atrium are contracting. So what we're talking about here is the ventricles. The atrium are contracted. They've pushed all the blood into the ventricles. The ventricles are relaxed. I want to know how much blood's in those ventricles. Like once the atria pushed all the blood into the ventricles, how much blood's in the ventricles? That's what end diastolic volume is. At the end of this whole thing, what is the volume of blood in the ventricles that are in diastole? It's never going to be 130. That's just a number that your book gives. You know, it's roughly, just to give you some ideas. Right, so it's not important that end diastolic volume is 130 mils. What's important is what is end diastolic volume. It's the amount of blood in the ventricles when they're relaxed. I know we're putting it here in atrial systole, but that's where it's appropriate. Because right now the ventricles are relaxed. So you've got three main ideas here. Blood filled the atria, opened the AV valves, and dropped into the ventricle. That's idea one. Idea two, the atrium contracted and pushed the rest of the blood into the ventricles. That's the second part. And then the third part, now how much blood's in the ventricles now? That's the end diastolic volume. So that's the first part of the cardiac cycle. Any questions on that? Now we're getting to the second part. Now it's the ventricle's turn to contract. So let me back up to small. All right, fine. They did a really shitty job on color coding for this whole thing. Because look, it's like why is the blood green? Like, I mean, I get what they're saying. Like, here's the blood in atrial systole, and it's filled up now, and it's opening the AV valves, and it's all gonna dump into the ventricles. That's what's happening here, and then the atrium are gonna contract and push the last bit into the ventricles. I don't know, but anyway, let's look at this bottom one. It might not. So when the ventricles contract, notice the ventricles here and notice them here. See how it's all squeezed? Here it's not. It's like the heart looks, the apex of the heart, the bottom of the heart looks a lot different here. You can tell it's squeezing, right? And so that blood wants to leave, wants to go out. You've got, each ventricle has two doors. There's a door here, that's your AV valve, and then there's a door there, that's your semilunar valve. Same thing here. There's a door here and a door there. So if we look at this, you've got two doors. Each ventricle has two possible doors. The AV valves are going to close because that door doesn't open into the room. It doesn't open into the ventricle. It opens into the ventricle, not out. Right? So the door is not going to swing the other way. So when that blood tries to go back into the atrium, it cannot do it. It closes the AV valve. Instead, this I don't know if you can see it, it's at like the tip of my pointer. The semilunar valve is going to open up and the blood's going to exit that way on both sides. So because the ventricles contract, the AV valves close and the semilunar valves instead, they open and the blood exits the heart. That's actually not too bad. Ventricles are contracting, aka systole. Ventricles are contracting, the atrium are relaxing, diastole. AV valves are going to close because whenever blood tries to flow backwards, it closes the valves. Whether they're the AV valves or the semilunar valve. When blood flows back, when blood tries to flow backwards, it shuts the valves. Then we have this again, end systolic volume, not end diastolic, but end systolic. So I want to know when the ventricles contract, how much blood is left in the ventricle, because not all of that blood gets pushed out. Some of that blood remains. So you have end systolic volume. And if you see down here, I added it in here. This is what stroke volume is. Stroke volume is essentially saying how much blood are you pushing out of your heart in every heartbeat. So that's the point of your heart beating, to push blood around your body, right? So I want to know how much blood is leaving your ventricles each time they contract. And to do that, we take both of these numbers, both of these readings. How much blood's in the ventricle when it was relaxed? How much blood's in the ventricle after it contracts? And then whatever the difference is, that's how much got pushed out.
So that number is right, 70 milliliters?
Yeah, 70 mils around there. Okay. 60 is something. It could be more, it could be less. Sometimes they'll say like, they'll use another reading called like an ejection fraction. 65 is fine. You don't want a little bit, like when your heart starts to get all messed up, your stroke volume is going to go down. It's instead of being 60, it'll be 48 or something. And it's like, okay, well, you're not pushing out as much. And if you think about it, if you get a heart attack, for example, you're going to mess up. You can mess up cells, like those cells don't repair themselves. So like when they're gone, they're gone. And so now you have a group of cells not being able to contract. Well, if part of your ventricle is not going to be able to contract, you're not going to push out as much blood, right? So your end systolic volume is going to be higher. There's more blood remaining in your ventricle because you didn't push as much out. There's lots of reasons. Of course, there's lots of reasons why it could all happen. But, and then we actually don't, we take stroke volume. You can't just, that's one heartbeat, right? You've got to take, we call like a cardiac output. You got to take it over, many heartbeats. You can't just take one heartbeat, right? So I don't even know if I have it on here. I have it here. We'll get to that. You take it like over a while. So I put stroke volume in here. I put it as part of ventricular systole. Your book treats it separate, but I'm just trying to. So, in short, ventricles contract, atrium are relaxed, the AV valve's closed, the blood gets forced out of the heart. Whatever blood's remaining is called end systolic volume. I don't care about the numbers of stroke volume, because they're just ideas. It's never going to be perfect like that. Oh, this person has a end diastolic volume of 128. So what? So I just want you to know what stroke volume is.
One more time. So the AV closes, and that's basically it forcing the rest of the blood out. And then the SL valve opens, right?
Yeah, imagine you're in a room and you've got like 30 people in there, right? And your room has two doors. One door is like a regular bedroom door. The door opens into the room. So that door is open, right? But it's not open like all the way. It's just open enough for people to come in and out. You got another door that opens out. You turn the doorknob and you push that door out into the hallway, right? So now everyone's going to run out of the room at the same time. They're going to go to the door that opens into the room. Everyone's going to smash up against that door and close it, right? And then it's going to be like at the stage at a concert, right? Everyone's just crowded up against it, and that door is not getting open because you all try to get out at the same time. Instead, go to the other door that opens out, right? If everyone smashes up against that door, that's fine. It opens out. It opens into the hallway. You're still going to be able to get out. That's kind of like your AV valve. It opens in to the room, so you can't. When you try to go out, it shuts it. So you go to the semilunar valve. That door opens out, so all the blood can leave through that way. If you watch an animation of the heart working, I think within a few minutes you see it all work. You're like, oh yeah, I get it. Because it's really a lot. It's one of those things that if you watch it work, then you get it, right? If I'm trying to explain it in words or reading a book, it makes it kind of harder. But once you see it, then you're like, I get it, right? Even if you see a regular heartbeat, you're like, okay, I get it. Because the top part of the heart is squeezing, then the bottom part of the heart is squeezing, and then the whole thing is relaxing. You're like, there's the 3 stages: atrial systole, ventricular systole, relaxation. I'm not making it worse by breaking it all down.
No, I don't think you're making it worse by breaking it down. I think it helps what the stage is. It's just, like I said, I would go look at a video to see it.
And the problem is that when you type in cardiac cycle, you're going to get some, because it can be way more complicated than what I'm putting here. And then that's what some of these books will do. If you see, some of them are for studying for the MLEs. And I mean, that's more information than we really need. So, and you might read in your book some stuff about isovolumetric. Let me make sure I don't have it. Oh, I do. It's all right. You'll read about isovolumetric relaxation and isovolumetric contraction. It's that's, I mean, it's kind of important, but it's not necessary. And it's there's a lot of details that just end up throwing you guys off. So I just think it's better to start simple and just see it see the blood moving through the atrium and ventricles, and you'll get it. You'll understand it. It'll make sense to you. Relaxation is when the whole heart's relaxing. The atrium and the ventricles, nothing's moving, right? There's a period of your heartbeat where that happens. When that's happening, blood is still flowing because you just got done pumping blood. It's still going through your body, right? And if you look at how blood flows through the veins, it's just kind of constantly flowing. If you look at your artery, it's pulsating. Like that's why you feel a pulse, right? It's moving like that. It's like jerking a little bit, a little bit, a little bit. Every time your heart beats, it just like jerks through your body. That's what you're feeling with the pulse. You're pressing on your artery that's close to like a bone, right? So that you can feel it when the blood surges through your artery. I don't know why I keep pointing here. So, but once it gets to your veins on the return trip to your heart, it's not pulsating anymore. It's just flowing. And what's my point? That blood's constantly moving. It doesn't just like stop. It's constantly going through your body. So even though your heart's in relaxation, the blood's still flowing back into your heart like normal. That's like a constant movement. So blood is flowing into the atria, and I put here, the pulmonary veins. You remember what side? If I'm talking about pulmonary veins, what side of the heart is that going to go into?
Is that the left side?
Left side. Vena right side. The AV valves are closed for a while because the blood's filling up, right? But eventually it's going to pop them open, right? But that's the next cycle, right? But now let's think about the ventricle. The ventricle's relaxed. Blood just left the ventricle. As soon as the ventricle relaxes, that blood turns around and tries to go back into the ventricle. It won't. It closes the AV valve. So like, look, here's an AV valve. Semilunar valve. Here's a semilunar valve. Here's a semilunar valve. So let's take this one. The blood goes out. The ventricle contracts. The blood gets pushed out. Then when the ventricle relaxes, that blood tries to flow back down. But it won't. It's going to shut that semilunar valve. That's actually the way your heart gets. Look, the valve's like right here. So imagine there's a semilunar valve right here, but you can't see it on this photo, right? And so blood gets pushed out, and then during relaxation, that blood tries to go back. But this valve shuts, and it keeps it from flowing back in. Instead, what it does is that it goes into this vessel right here. So every time your heart relaxes, you're going to get blood going into these coronary arteries. So that's going to that's how the heart feeds itself blood. So the semilunar valve's shut. You can like barely see it underneath there. It shuts, and the blood can't go back in. That's what you hear with a heartbeat. Your heartbeat, you're hearing like, it actually makes 4 sounds, but you're hearing like 2. That first sound is the AV valves closing, and the second sound is the semilunar valves closing. But you notice there's like there's a break in between. So it's not like constant. That's not how your heart sounds, right? It's because there's a relaxation stage. There's a part where the heart's not doing anything. All right, let's get back to the top. Relaxation. Blood's filling the atrium and the ventricles. Blood's filling the atrium. The blood that just left the ventricles tries to flow back in, but it won't because the semilunar valves close. So actually, you've got a point here where all four valves are closed. The AV valves are closed because you didn't get enough blood in the atrium yet. It's not like heavy enough to pop those AV valves open. But the blood tried to flow back into the ventricle and it closed the semilunar valves. I don't care about isovolumetric relaxation. You could skip that last one. It's fine with me. Because it just probably throws in more confusion. ISO, just FYI, I don't know what it means exactly, but I think it means like the same. Isovolumetric meaning like the same volume, or like an isotonic solution has like the same amount of like an IV bag is isotonic. It's got 0.9% sodium chloride. So does your blood. It's equal. Maybe iso means equal, something like that. That's just FYI. So 3 parts to this: atrial systole, ventricular systole, relaxation. I will ask you this. Explaining it will be, I mean, I don't want bullet points. I'm trying to get you to explain it.
So, like for the relaxation stage, you want us to know that the atrium and the ventricle they're both at diastole, which is relaxation.
They're both what?
But blood is still circulating even when the atrium and ventricle is relaxed. The blood is still circulating, and then when it gets pushed down, the AV closes. Like, I guess transitioning into that part is where I'm kind of confused. Like, the blood is still circulating, that's what you're saying, and then it goes down to gets forced out through the AV valves and they close.
Yeah, okay. Part of that is my fault messing you up.
No, I don't know.
You got I guess the arteries and veins part is not like so important. Just know that blood is filling. It's not like blood stopped in your body just because the heart stopped beating for that fraction of a second, right?
Right.
It's not like all blood stopped. If I were to look at your artery, the blood stops. As it's moving down my arm, it's going like that, right? It's just every time the heart beats, it moves a little further. But if I look at the veins coming back, it's just going like this. It don't matter if the heart's beating or not beating. It's just continuously moving.
Okay.
So blood in your arteries and your veins, they move different. When you cut a vein, blood's like a little bit darker and it just kind of flows out. Right? It looks dramatic, but it's not. When you cut into an artery, it spurts. Like, it goes 5 feet. Maybe I'm being dramatic, but it goes far. Right? So when you see people's like shunts or something, like, the blood doesn't just go like that. It's gonna hit you if you're standing like in front of them. Like, the blood will hit you. So blood moves different in an artery because it's really pressurized. It moves different in an artery versus a vein. So when a vein is continuously flowing. So my point is, when your heart's relaxed, blood's just it's just filling up it's still filling up the atrium. I shouldn't have explained all that because I think I just made it worse. But blood's filling the atrium. Just know that, right? The whole heart's relaxed. Atrium, ventricles are relaxed. That's what I'm saying here. Blood's still filling the atrium, though, even though it's relaxed. The AV valves were still closed from the last cycle, right here. Because the ventricle, when the blood tried to go from the ventricles back into the atria, it closed the AV valves, so they're still closed. But the blood that you see here, semilunar valves were opened, and when those semilunar valves opened, the blood left the heart. As soon as the ventricles relax, that blood tries to return, tries to backflow. So it's going to close those semilunar valves.
So this process is basically what we're hearing if we put on a stethoscope and put the cuff around someone's arm, and then when we're hearing, when we're pumping, and then it starts to come down, and you hear that ba boom, ba boom, ba boom.
Yeah.
This is kind of this is that process, right?
It is. It is. It's what
Because now, yeah, because I mean, nowadays everyone has a machine, but in like this program I went to, they show us how to check it with our stethoscope, and you put it on the on the cuff part of your wrist, and then you listen, and it goes ba bump, ba bump, and then there's a point where you can't hear anything, and then that's when you start to hear something, and I think that's how you get the diastolic. If I'm thinking right, I don't know. But
doctors can, like especially cardiologists, but like doctors are really good at like they can listen to the valves in your heart, and they can hear them close if they're closing the right way, or if they're not closing, or if they don't quite, like they're supposed to close like this, but maybe something happens where like your atrium stretches a little bit. So when they close, just imagine the bridge closes, but it doesn't like one of the one bridge closes all the way, but the other one doesn't, right? So blood can get back in, right? They can hear that, like they can hear that with a stethoscope. They can hear like abnormalities in your valves, and if this whole cardiac cycle is not working like it should, like they can hear it just by like the sounds that your valves make. So yeah, that's what they're listening for. Cardiac output, I was saying, stroke volume is how much blood gets pushed out of your ventricles every time they contract, but you don't take one reading, right? So cardiac output is stroke volume times however many beats per minute, right? So you take how fast your heart beats, and you take stroke volume. So not that I want you to know numbers, but let's say stroke volume 60, and let's say your heart's beating 60 times a minute. So you would just do 60 times 60, and that would give you 3,600. So that would be cardiac output, 3.6 liters. So like every minute, your body's moving around, your heart's pushing out 3.6 liters. It just know that cardiac output is stroke volume, and then times the beats per minute.
What does stroke volume mean again?
Stroke volume is how much blood your ventricles are pushing out each time it beats. It's like the key to heart health. If your heart is healthy and working like it should, you should be pushing out a certain amount of blood. I mean, that's the whole point of your heart, is to circulate blood around your body. So I want it to pump. Every time it beats, I want a certain amount of blood coming out. That's stroke volume. That's each time it beats, how much blood's coming out. Of course, I need to know that over like a minute. I don't want to just see one beat. Maybe you're, it's like taking one test and getting a grade for the entire semester. We need to. And cardiac reserve is this is just showing you what we're capable of, right? We're capable of putting out 4 times more than just regular cardiac output. Most of that is going to be because your heart beats faster, right? Your heart's beating, let's just give a number, 70 beats per minute, right? But that could jump up to 200. We could almost triple your heart rate. So obviously that means you're putting out more blood, right? But the body also becomes better at, or it's also able to like push out more stroke volume. Like, it doesn't matter. It's able to push out more if it has to. You think your ventricles are squeezing and pushing blood out. Things like adrenaline make them squeeze even harder, which would push out more blood, right? So all I'm saying here is that you have the ability to push out like at least 4 times as much blood if you had to. So we call that cardiac reserve. How much blood are you pushing out per minute right now just sitting versus if you got adrenaline flowing through you. I'm not going to test you on this. I'd like you to know what cardiac output is, but I'll probably roll that into a different question. Cardiac reserve, I'm not going to ask you. So we got 4 questions so far. 1, cardiac cycle. 2, EKG. 3, however I call this. I know that kills you when I say that. I'll call it, I don't know, action potential. How is an action potential generated in a cell, in a muscle cell? 4, cardiac conduction cycle or cardiac conduction. So we got 4 things we've talked about. Try to squeeze in one more thing. I need 5. I might do a separate question about what is stroke volume. I don't know. Probably not though. Right. This is question 5. Actually, we were just talking about this. This is question 5, regulation of stroke volume. Stroke volume is how much blood gets pushed out of the heart every time the ventricles contract. Right? So there's different things we can, that besides speeding up the heart rate, this has nothing to do with that. There's different things that you can do to push out more blood every time you contract. One's called preload. If the ventricles stretch, the more it stretches out, the more blood's going to be ejected when you contract it. Right? So you want your heart to have some stretchiness to it. It's like a balloon, like a water balloon. And you're gonna take the end of it. You're not gonna tie the end. You're just gonna kind of not hold it and let it spray out, right? The more water you can get into that balloon, the more you can spray out of it. No, that's not a good example, but that's what preload is. So the amount of heart that the heart can stretch before it contracts. It's important because with some people, their heart becomes less stretchy. Like the elastic becomes like less elasticy. It's not a word, but less elastic fibers, right? And so that's going to affect this. You can't stretch out your ventricle as much before you contract it. Contractility is just what I put, the forcefulness of the contraction. That's what norepinephrine does. We've always talked about epinephrine, but just FYI, norepinephrine makes it squeeze even harder, right? It contracts harder. So the harder you contract the ventricle, the more blood's going to come out. The third one's difficult, afterload. You might just want to memorize it rather than understand it, but what it's saying is that when you squeeze a ventricle, you're eventually going to make the valve pop open and all the blood's going to leave, right? So this is what afterload is. How much can you squeeze it before that valve pops open and everything just leaves? The more you can squeeze it, the more it's going to leave. So the more pressure you can build up in that ventricle before the valve opens, the better. Right? You don't want the valve, you don't want like a weak ass valve that just, as soon as you apply some pressure to it, it just opens up and then everything's just going to kind of trickle out. You want that valve to hold close, hold, and then bam, pop open, and that blood just flies out. That's what afterload is. So that's question 5, regulation of stroke volume. This is my very last question. So I'm going to get 6 and I'm going to ask 5 out of them. So you're going to have all the questions here. Oh, I don't even have a slide for it. 6 is talking about, I think I've got a better, like in the other lecture I gave, I think I wrote this out on a slide, but this is about regulation of heart rate. how do we regulate our heart rate? How do we make our heart beat faster or slower? The first method is our brain, the neurological, the nervous system, right? Do you remember sympathetic versus parasympathetic? I was telling you sympathetic is fight or flight. And I was using the example of, I don't know, somebody throwing a grenade in a room or something, right? That's fight or flight. Or there's a snake in your room, right? You could kill the snake or run away from the snake. That's fight or flight. That's sympathetic. People think of sympathetic as like the opposite, but it's that's sympathetic nervous system, fight or flight. Everything else you do is parasympathetic. So sleeping at night, digesting a nice meal, that's not what we're talking about. We're talking about sympathetic. Looking for the word in here. It's not even in here. Why am I spending so much time on it? Oh yeah, it is. All right, so there's sympathetic, right? So you have a cardiovascular center. That's like the central computer of how fast your heart's going to speed up or not, right? So that's in your brain stem. Not that you need to know it, but it's like the medulla oblongata. So in your brain stem, you've got like the center. You've got nerves hooked up to that from around your body. So in your body, you've got these different receptors, and they're listed right here, and I'd like you to know what they are. So you have proprioceptors, chemo, and baro. Chemo means chemicals, so it's monitoring chemicals in your blood. For example, it's looking at carbon dioxide. Do you have too much carbon dioxide in your blood? Okay, I'm going to probably speed up your heart rate. I'm going to make you breathe faster. I want to try to get that carbon dioxide out. I want to get it to your lungs fast, and I want to get you to breathe it out. So the chemoreceptors are picking that up. Too much carbon dioxide. Baro means pressure, like a barometer. Baro means pressure. Is your blood pressure too low? I'm going to send a signal to your cardiovascular center and tell your brain, hey, go tell the heart to speed up its heart rate. The pressure is low. Right? So that's the signal in, and that's the signal back out to the heart. So you've got a wire going from different places in your body up to your brain. You've got another wire coming back down to your heart. Speed your heart up or slow it down. The third one's proprio. Right? It's monitoring limb movements, right? I got both my arms above my head, and I've got my legs up, right? I need to, since my arms are above my head, I'm triggering my proprioceptors. They're telling my brain right now, this dude's holding his arms above his head. Can you like speed the heart rate up a little bit? Because we got to get blood up to his hands now because he's an idiot and he's holding them above his head. And now they stopped. Now they went back again. Now they stopped. So that's proprioceptors. They're monitoring what I'm doing with my arms and legs, and they're sending signals to my vascular center, and my cardiovascular center is hooked up to my heart, and it's making my heart beat go faster or slower. Vagus nerve. Well, you know what? Don't worry about it for now. But that's what it is. Right, and so that can make things happen right away, right? And then you also, of course, you have, things that your eyes see, things that your ears can hear, things that you smell. That's all linked up as well, and it just makes your heart beat faster or slower. I don't know how to explain that, but I don't know how to put it on a test other than just like explain it like that. Yeah, so what they're doing, the SA node is the pacemaker of the heart, right? So they're increasing stimulation to your SA sinoatrial node, and that's what's making your heart rate go faster. And look what else it can do. It could increase stroke volume. So decreasing contractility is one of the 3 factors affecting stroke volume, right? So what they're saying here is increase heart rate and increase the amount of blood that gets pushed out of your heart each beat. So there are some chemicals. I don't have the chemicals mentioned here, but you already know what they are. Epinephrine, norepinephrine. Those are going to speed up your heart rate and speed up stroke volume. So epinephrine, norepinephrine, it's the same word, epinephrine, just put N-O-R. So same word. That's, adrenaline. So those are hormones that speed up your heart rate. And I'm going to give you one more. AN as in Nancy P. Stands for atrial natriuretic peptide. ANP is a hormone that's made by your heart, and it slows the heart down. It's made by, it's when the atria get too much pressure inside them, it's going to try to decrease that. It's going to slow the heart rate down. So ANP slows heart rate down. So this is one question. And I've got I'm going to post a separate, smaller lectures. As soon as we're done here, I'm going to post a bunch of smaller lectures, and it will have more detail about this last question. That's one question. Cycle question 2. EKG question 3. This one question 4. Cardiac conduction system of the heart question 5. You guys get it. 1, question 2, question 3, question 4, question 5, question 6. 6 questions. I'm going to take 5 of them. So you know what all the questions are.
So we don't have to need to know the blood flow through the heart.
I want to say yes now. It's super important, but I don't want to punish you for asking that question either. How would you guys? Are you guys did you already study it and you feel like, oh, that's definitely an easy one, so put that on there? Because I could do that.
Wait, you talking about the lecture from last time?
Yeah, I forgot about it.
Don't even bring that up.
Okay.
What the heck?
Then no, the answer.
Got enough going on.
That was easy to me.
To you.
The first lecture was way easier.
To you. The first lecture had me this lecture is smooth sailing to me.
For real?
Well, it's memorization.
Yes.
It goes here, then here, then here, then here. It's like memorization. I'm good either way. I'm inclined to not put it on there because I've been talking about 6 questions. So I know a couple of you studied it. It's not like it's not gonna, you'll see it again on another test. All right, so I won't put blood flow to the heart now. I'll probably put it on exam 2. So we'll just stick with these 6 questions. I'm gonna pick 5 out of these 6.
Make it a bonus question.
Okay. All right, I will. It'll be a bonus question. There's no reason for you not to nail this. If you prepare your questions beforehand, you could. I know it's a lot of material. I'm not saying it's easy, but it's not something you don't know. It's just it's going to require work. All right, let me stop this recording. You guys have any questions?